Thank you for joining us today for our fourth webinar in a series that focuses on improving immunohistochemistry and immunofluorescence workflows. Today's webinar is about counterstaining in IHC. This will be presented by my colleague, Dr. Craig Powell. Following the 20-minute presentation, we will have 10 minutes for a Q&A session. Please feel free to submit any questions you may have using the chat function on the GoToWebinar site, and we will answer your questions accordingly. I am now pleased to introduce you my colleague, Dr. Craig Powell. Over to you, Craig.
Thanks, Byron. Hello, everyone, and good morning, good afternoon, and good evening, wherever you may be located around the globe. Thank you for joining us for this fourth webinar in our series. Now, our focus today, as Byron mentioned, is on counterstaining in immunohistochemistry. Now, just before I commence the webinar, let me just explain why we feel this topic about counterstaining is important enough for a webinar. Now, as most of you know, the counterstaining procedure is conducted right at the end of the IHC or immunohistochemistry workflow. Now, a lot of investigators work very hard to obtain the specimen for the tissue preparation, blocking it, cutting it, doing the immunohistochemistry to get a good stain. But all too often at the end, what we see is this particular counterstaining step receives very little attention, and the counterstain is applied haphazardly or dogmatically, and it really detracts from the specific staining of their specimen that they've worked so hard to get. So suddenly it would have, in many cases, a negative impact. So hopefully this webinar will give you some insights into how to optimize the counterstain and maximize your results for your immunohistochemistry specimen. Okay, without further ado, the brief overview will be what is a counterstain for those of you who may be new to the field, and we'll also look at why apply a counterstain in immunohistochemistry. Then we'll look at some commonly used examples of counterstains for immunohistochemistry, some main considerations when applying a counterstain, and then we'll go over a brief summary. Okay, so I've got a bit of a definition here for those who are new to the field in what is a counterstain, and I've got here a dye or a stain that binds to cellular components and provides contrast with the specific immunohistochemistry enzyme substrate precipitate. That may be fairly straightforward, but I believe this is a fairly elementary definition and doesn't really get to the point of why you may want to apply a counterstain or the value of a counterstain in immunohistochemistry. And indeed, I will say that one key facet of the counterstain is to augment the specific staining to generate additional information with respect to antigen localization, regional or spatial expression of that antigen, and the tissue morphology. Now, if you remember the example on the title slide, the same image here, and what we've got is this particular brown staining circle. It's actually a lymphatic endothelial cell marker, and what we've done is also counterstain with a blue nuclear counterstain here. If we did not apply this counterstain, all we'd have would be this brown circle floating in space and not give you any appreciation of its association with this given arteriole. So certainly in this single stain example, you can see the value of why you would want to use a counterstain for immunohistochemistry. Now, well, just keep in mind this particular image as we go forward actually through the webinar. I'll refer back to this image a couple of times. Now, I'm not a particular fan, I'll say, of the phrase counterstain, and I think this is probably a shortcoming a little bit of the English language, but to take a counter position is to take an opposing position or an opposite position from what is presented. And I don't like the idea of the counter stain being opposing necessarily to the specific stain. I like to think of them more as compatibility or companion stains to work in concert with that specific staining and maximize the data you can get from your tissue section. Okay, let's look at some of the common counterstain choices that are used out there for immunohistochemistry. And what I've got here is a short list of some of the hematoxylins that are used. Mayers, there's a modified Mayers, there's Gill's type 1, 2, and 3, Harris, there's a modified Harris, Ehrlich's, there are other hematoxins as well. And what I've got here on this image on the right is they all do give you blue nuclei, I'll use the phrase, at the end of the day. So any or all of these would certainly be suitable for generating this sort of image on the right-hand side that I've got here. This is not an exhaustive list. There'd be some considerations, though, as we'll mention later on, about certain formulations of hematoxylin. So just keep that in mind. Now, I've got some investigators who may not be particularly familiar with the techniques or some of the histology perspectives, and they tend to put on a hematoxylin and eosin counterstain in combination with their immunohistochemistry. Now, hopefully the image I've got here showcases that, there's just an absolute drowning of color in this particular image. And at least from my perspective, while you could apply a hematoxylin and eosin with some given antigen expression in immunohistochemistry, I really don't see there's a need for it. And the eosin could certainly work against some of the specific staining you're trying to identify. So I would highly recommend against using eosin as part of your counterstain, unless you had a specific reason for it. But if you just want to apply a hematoxylin, do that without the eosin. Certainly there is a place for hematoxylin and eosin in your immunohistochemistry, and it would be on a separate tissue section, just to look at some of the underlying tissue morphology and architecture, or give you a greater perspective on that outside of the immunohistochemistry. So it would be on a different tissue section. Here's some other counterstain choices that are popular out there as well. Methyl green, that gives you a light green nuclear stain, and a nuclear fast red that gives you sort of a pinky red nuclei. Now, these counterstain choices are not exhaustive by any means, but they're the most popular ones to use. And you'd have to probably have a fairly good reason of why you'd want to wander away from these or look at others like Giemsa stains or something like that. Again, not an exhaustive list, and others certainly do use and publish on using other counterstain choices. Now let's get into why apply a counterstain. And certainly the first question when considering that is, do you actually need a counterstain? I think that's a fair question to ask. And the example I've got here on the right-hand side is a triple label immunohistochemistry. We've got a cytokeratin marked here with a red alkaline phosphatase substrate. We've got a Chi67 highlighted here with some brown DAB. And then out here, I believe it's CD34 using vector blue. So in this particular example of immunohistochemistry, now there'd be no reason why you would add a fourth color. There's no value to that. Indeed, it may be problematic and would detract from that specific staining. So the question you want to ask is, does the counterstain add additional information, or does it actually subtract or negatively impact the specific staining of my given application? And sure, I've got an example here of a triple label. Think back to that title slide image, the one with the lymphatic endothelial cell marker, and indeed the arteriole. You can see the value of a counterstain there. So certainly a single label in some instances, or in most cases, would benefit from a nicely counterstained specimen. Now, some investigators, even if they are doing, say, a triple label or something like that, just follow through an SOP and dogmatically apply a counterstain because, well, we've always done it this way, or it's part of the SOP, or I was told to do it. Just have a look at the actual methodology you've got there and ask the question, is the counterstain going to be a benefit to me, or is it even required? So don't be a victim of lab dogma and be told, oh, apply the counterstain for a given period of time because you've always done it that way. You know, that's a bunch of nonsense. And quite frankly, if you want good staining, good results, just ask the question, do I need to use a counterstain? Here are some commonly used combinations of counterstains. Brown hematoxylin shown here on the left-hand side with a blue nuclei stained with hematoxylin. Brown DAB, excuse me, with blue hematoxylin. And over on this image here on the right, brown DAB toning for a different antigen in contrast with a nice methyl green counterstain. A couple of other examples here. On the left-hand side, Vector VIP, it's a purple color in contrast with the methyl green. And over on the right-hand side here, a smoky bluish gray deposit here using Vector SG in combination with a nuclear fast red or this pinky stain. Some of those who may have eagle eyes out there will also see this third color, which is actually melanin deposits. This is actually a melanoma preparation. So those of you who might be looking for alternatives to what you're currently using, or to mix it up a little bit, there are certainly a number of different combinations you could use. These examples here are just to give you an idea of what is possible, what is acceptable, what gives you a great contrast out there. Not exhaustive by any means, but just gives you some insight into other color combinations. Okay, this gets us into what I see as a key issue that some investigators encounter when applying a counterstain. Now, what I've got here are two sections that just happen to be serial sections of breast carcinoma that have been stained using immunohistochemistry for estrogen receptor using brown DAB. Now, estrogen receptor is a nuclear expressed antigen, and so it does deposit in the nucleus of cells, and certainly hematoxylin also is a nuclear counterstain. So what we do see quite often, or what we encounter from investigators, is a scenario such as the one shown here, whereby we have a very robust or a heavy counterstain that masks or obscures the specific stain of the estrogen receptor. And unfortunately, this would be interpreted as a false negative. You have to be very careful with counterstains in this situation, and you want to get more of a light or a balanced counterstain, such as the one that's shown here, whereby you can differentiate the brown estrogen receptor localization in contrast with the other blue nuclei. And certainly, you know, from the Ventana Roche, what the term they use is visual acuity. So you've got to aim for this nice light balanced counterstain that works in concert or parallel with your specific stain. So be cognizant of the localization or the intensity of the target antigen staining. Certainly this is one scenario that plays out quite often that we've encountered with end users. So how do you get from the left image to the right image, or what do you do? What are the parameters to consider? And certainly have a look at the length of time that the specimen is exposed or incubated in the counterstain. There may be pH differences as well, and also the use of a differentiator. I'm not going to get into the weeds, so to speak, on the use of progressive or regressive counterstains at this time, but just be aware that there are differences in the counterstain. And what I'd highly recommend for any new tissue block that you are starting to cut into or look at is to run a negative control, whereby it is just for counterstaining only. A tissue section that's been freshly cut and exposed to your counterstain of choice, whether it be hematoxylin or methyl green or whatever, and try and get this light balanced look or counterstain appearance before wasting valuable time and effort with your immunohistochemistry. So just make sure you run that defined negative control section. Now, if you remember back earlier on, I did describe a little bit about some of the various formulations of hematoxylin that are available. And I've got here an example of Harris hematoxylin and a Mayer's hematoxylin. And I did say they do stain the nuclei blue, and at the end of the day, sure, they would all be suitable. But just be aware when you are applying some of these counterstains that some of them may contain solvents such as ethanol or alcohol that may have a detrimental effect on the specific substrate of choice that you're using. It may affect solubility or intensity of that given stain. And so if you do look at that under the microscope prior to applying the counterstain, you may say, hey, this is really nice specific staining I've got here. But after the counterstain, it may look a little duller or washed out, or indeed, have suffered some sort of washout effect. So in these cases, I'd highly recommend going with more of an aqueous-based counterstain recipe. Okay, some other considerations for counterstain: specimen fixation and preparation. Certainly the type of fixative can influence the uptake of a stain. Now, a lot of these parameters, and I'll say maybe the fixation and the blocking of a specimen, may be outside of your control, particularly those who may be working in core facilities or a CRO. You may simply be handed the material and be asked to work up an immunohistochemistry stain and counterstain it. It's certainly imperative at that point, working with an unknown, to run that defined negative control of just a counterstain alone, just to get a feel for that intensity of the counterstain. Also, the mode of fixation as well can play into uptake of stain and the appearance of it. Now, if you remember back to that original slide again, on the title slide where we had that lymphatic endothelial marker, that brown circle with the nicely stained adjacent arteriole, those of you who may be familiar with some histological applications would be familiar with the fact that that would be an immerse fixed specimen, something that was excised and placed into fixative. If that was perfuse fixed, then that particular arteriole would be much thinner walled, and the nuclei that you'd be targeting with the counterstain would be more spindle-like, they're squished, if you will, and that would affect the uptake of the stain. So just be aware also of the mode of fixation when you go to look at it, and also when you run that negative control. The thickness of a specimen in terms of the micrometers is also important as well. And I'll just draw from a real life example, just from a couple of weeks ago. Some investigators from Cornell called up. They were using fresh frozen materials, cutting the sections at about 20 microns. What they were doing, cutting the section on a cryostat, mounting it onto a glass slide, performing fixation with paraformaldehyde, about 2 to 4%, and then immediately applying a counterstain. This particular material had not been exposed to xylenes or ethanols or anything like that, like a paraffin embedded section, and they weren't getting uptake of hematoxylin. And they're scratching their head saying, what are we doing wrong? Can you help us out? And basically they had to address some permeability issues there. So we described putting on some Tween 20, Triton X100 detergent, and that significantly added to the uptake of the stain. So just be aware of the thickness of the section, and that can certainly play a role in the effectiveness of the uptake of the stain. Tissue type also factors in. I know a lot of investigators simply don't work on one type of tissue, but if you're working on, say, the example I've got here is lung tissue, you also want to look at the expression of the same antigen in kidney tissue. Take a step back a minute and just understand that these will be different tissue types, and it will factor in with how the counterstain appears on that given tissue. So run that negative control. This is also a factor if you are working between species as well. Certainly, I've got an example here of an amphibian species. You're working on skin or cardiac muscle with that amphibian. You want to swap to a mammalian species. Again, more than likely that counterstain will be a little different in the way it does behave on that different species tissue. So again, just be aware of that, and all things will not be exactly the same. So work it up on that negative control. The method of application certainly does factor in as well. And what I've got here on the left-hand side is a slide, just showing a manual application here. I'd say this is particularly important for counterstains that do require heating. Example here is methyl green. Some investigators put a slide like this on a heat block, then apply the methyl green that requires heating to 60 degrees, and then they take it off after a given period of time and notice that there is no sufficient uptake of stain. For something like this, I'd highly recommend a batch staining where you put the slides in a slide dish, submerge them in a volume of heated counterstain, 300 to 500 mLs, so you get uniformity and consistency across all of your slides. Now, certainly if you're working with any more than, say, 3 slides, I would highly recommend using this batch staining method as well to get that consistency and uniformity as well. Just be aware of the fact if you are changing between counterstains, you want to mix it up. Not all of them are the same. As I've just mentioned, methyl green requires heating. That's not a requirement of hematoxylin or nuclear fast red. So again, don't expect methyl green to look or behave exactly the same as hematoxylin or fast red. Be aware of the fact too that these do have shelf lives. Methyl green, for example, should be a nice deep dark forest green color. If it has tinges of brown or rusty brown, certainly it has expired, so you will have to get fresh material. So a quick summary here of some of the key points that hopefully will help you get some successful counterstaining. Number 1, establish that a counterstain would be beneficial in your given specimen. Secondly, select an appropriate counterstain and substrate color combination. We did show you some. There are many others you can choose from. Understand the target antigen staining pattern and expression level. If you're looking at a very weak nuclear antigen, obviously you're not going to drown it in the counterstain. You need to get that nice balance and visual acuity. This is where you'd want to run a defined negative control and work up those defined staining parameters. As we just mentioned, any more than say about 3 slides, I'd highly recommend using a batch format for consistency and uniformity across all of the sections or slides that you're working with. And certainly use fresh or non-expired material, obviously, for the best results. For those of you who may be interested in learning a little bit more about some counterstain substrate combinations, we do have a free poster here. You're more than welcome to access to sign up to get through this particular URL. This gives you an idea of the dimensions of the poster, but we do have some valuable information down here about substrate combinations and substrate counterstain combinations. So at this point, I'd like to say thank you very much for staying with us for this webinar. Please tune in for our next webinar, Considerations for Selecting IHC Detection Reagents, on October 6th. So thank you again, and at the moment, right now, I'll open the floor up to any questions you may have. Thank you.
All right, thank you, Craig, for that presentation. We will now begin our Q&A session. Okay, so our first question is about methyl green. How can I troubleshoot light or weak methyl green staining to improve uptake?
Yeah, methyl green, as I did mention briefly in the presentation, it is certainly different from something like hematoxylin. It does require heating, usually to say 60 degrees is the recommended temperature range. And one of the issues I did mention is about investigators who may have a heating block handy, and they actually put the glass slide down on the heating block that's set to 60 degrees and then apply the counterstain right on top of that. And that unfortunately does not generate a sufficient uptake of the stain. So number 1, I'd highly recommend heating a volume of that counterstain to, either in a Copeland jar or in a staining rack or dish, 300 to 500 mLs, I think I mentioned. Heat that to 60 degrees, then submerge the specimen in that preheated solution. Additionally, methyl green in the methodology also has some steps washing through acetone, acetic acid, if I remember correctly. Those can certainly be reduced in time, so you can reduce the exposure to those, or indeed in cases of very weak staining, you can even omit those just to retain maximal methyl green staining. So hopefully those couple of points will certainly help you out. Oh, the other point I'd have to say is just make sure that it doesn't look, you know, brown or off colored, and that does have that, the stock solution has that, you know, dark, deep green color to it.
All right, thank you, Craig. Okay, the next question is about automated IHC. So they use automated IHC and their counterstaining is not always the best. Do you have any suggestions to improve on this?
Yeah, I will say I did really not touch on automated staining in this given webinar. Certainly that may be particular to, well, things you can certainly determine at your end. Maybe run it manually initially on a couple of different tissue sections just to gauge the parameters, as we mentioned, things such as time or whether or not it may be differentiator required, or indeed the actual formulation of the counterstain you're using. Make sure it might be one I'd recommend, as with an aqueous-based media, maybe just run it manually and then see if you can get those same determinants, same parameters set up on the automated stainer. So they would be things to consider. But, you know, I think that may be particular to the choice of counterstain and maybe the methodology of how the counterstain is also applied in that automated stainer. And I'd have to say that, unfortunately, that's beyond the scope of this given webinar.
All right, thank you. The next question is about hematoxylin. What is the optimal differentiation step for hematoxylin, acetic acid, or HCl?
Yes, certainly I do know that, at least in the formulations of some of the different hematoxins, there are different acids that are used. But the different differentiators, you know, may be particular to the actual formulation of hematoxylin you're using. So I don't know that there's a universal answer to that. Indeed, I would highly recommend looking at the formulation or the recipe of the hematoxylin you've got there, and certainly use an appropriate differentiator for that. And this is where it'd be important to run that negative control section, that test section, just to gauge some of those parameters. Look for consistency. Make sure that you run that one or two test sections for each different tissue type and certainly species of tissue that you're working on, just to gauge that intensity that you're looking for, that you will find appropriate for that given application.
All right, thank you, Craig. The next question is, what are the major differences between regressive and progressive counterstains?
Yeah, progressive stain tends to be, the longer you leave it in there, this is painting with a very broad brush, the deeper stain you will get. And I'll refer back to the couple of sections I showed of breast carcinoma, whereby the very heavy or robust staining we showed in the image on the left was a hematoxylin progressive stain that had been left in there for a considerable period of time. So certainly with a progressive stain, you can gauge it by leaving it in for a short period. If the staining is not intense enough, put it in again, and obviously as you move forward or progress like that with the length of time, you get a deeper, more robust or heavier stain. A regressive stain is usually something that's overstained, and then you use the differentiator to hone back or peel back the intensity to the desired contrast that you want. So painting with a broad brush, that's the sort of key differences between progressive and regressive counterstains.
All right, thank you, Craig. The next question is about hematoxylin. So my hematoxylin counterstain is very dark and shows poor contrast. I've already decreased the incubation time down to 10 seconds. What else can I do to troubleshoot?
Wow, okay. Certainly the concentration may factor in as well. Just make sure, and I'm not sure if you are making the hematoxylin yourself or indeed buying this from a commercial source. Just to let you know, the example I used there of the gills formulation, type 1, type 2, type 3. Type 1 is like a single strength hematoxylin. Type 2 is like a double strength. Type 3 is like a triple strength hematoxylin. So certainly the concentration of hematoxylin there, if you are using, say, a gills, very strong, very quick uptake of the stain. You may want to look at a different formulation of hematoxylin, such as like a gills 1 or indeed another type of hematoxylin that would help you. Another opportunity would be possibly using a regressive type hematoxylin, whereby you can control the intensity by then applying that differentiator and dialing back that intensity, almost like washing it off until you get that desired intensity.
All right, thank you, Craig. Our time is up for today for this webinar. Please feel free to join our next webinar October 6th about selecting IHC reagents. We will send you the entire recording of the webinar after this session. And if we did not answer your questions, we will answer them personally later. Please feel free to submit any more questions through our general email, technical@vectorlabs.com. Thank you for joining us and have a great day.
Thank you very much.